Health and Disease Introduction, 2nd PUC NEAT. Human health refers to a state of complete physical, mental, and social well-being, and not merely the absence of disease. Good health is essential for normal body functioning, efficient work, productivity, and a long lifespan. Health is influenced by genetic factors, infections, and lifestyle, including diet, exercise, rest, and personal habits. When the normal functioning of one or more organs or body systems is disturbed and signs and symptoms appear, the condition is called a disease. Diseases are broadly classified into infectious diseases and non-infectious diseases. Infectious diseases are caused by pathogens such as bacteria, viruses, fungi, protozoans, and helminths, and can spread from one person to another. Non-infectious diseases, such as cancer and genetic disorders, generally do not spread from person to person. This chapter mainly deals with 1. Common diseases in humans. 2. Human immune system and immunity. 3. Vaccination and immunization. 4. AIDS and cancer. 5. Drug and alcohol abuse. NEAT focus. For NEAT, this chapter is highly important because many questions are directly based on NCRT facts: disease-causing organisms, symptoms, modes of transmission, immunity, vaccines, and disorders. So, read the NCRT chapter carefully and remember important examples and diagrams. Diseases in humans. NEAT 2nd PUC Biology. This is an important part of human health and disease. Diseases in humans are caused by different pathogens, including bacteria, viruses, fungi, protozoan, and helminths, worms. These pathogens enter the body, multiply, and interfere with normal body functions, causing disease. #1. Typhoid. Causative organism Salmonella typhi, a bacterium. Transmission. Through food and water contaminated with the bacterium. The pathogen enters the small intestine and can spread to other organs through the blood. Symptoms: sustained high fever, 39 to 40 degrees Celsius. Weakness, stomach pain, constipation, headache, loss of appetite. Severe complications, intestinal perforation, may cause death in severe cases. Diagnosis: Whittle test is used for confirmation of typhoid. NEAT point typhoid, Salmonella typhi, Whittle test. 2. Pneumonia, causative organisms, Streptococcus pneumonia, Haemophilus influenza, target organ. The infection affects the alveoli of the lungs. What happens? The alveoli normally contain air and are responsible for gas exchange. During pneumonia, the alveoli become filled with fluid, causing difficulty in respiration. Transmission: inhalation of droplets or aerosols released by an infected person, sharing glasses and utensils with an infected person. Symptoms: fever, chills, cough, headache, difficulty in breathing. In severe cases, lips and fingernails may become gray to bluish due to insufficient oxygen. NEAT point pneumonia, alveoli filled with fluid, breathing difficulty. 3. Common cold. Causative organism rhinoviruses. Site of infection: nose, respiratory passage. Important: rhinoviruses generally infect the nose and respiratory passage, but not the lungs. Transmission: droplets from coughs and sneezes. Contaminated objects such as books, cups, door handles, keyboards, etc. Symptoms: nasal congestion, nasal discharge, sore throat, hoarseness, cough, headache, tiredness. Usually symptoms last for about 3 to 7 days. NEAT point: common cold, rhinovirus, nose and respiratory passage, not lungs. Malaria is caused by a protozoan parasite. Causative organism: Plasmodium. Important species: Plasmodium vivax, Plasmodium malariae, Plasmodium falciparum. Most dangerous species: Plasmodium falciparum causes malignant malaria and may be fatal. Transmission of malaria: the vector is the infected female Anopheles mosquito. Life cycle in humans. Step 1. Entry of parasite. When an infected female Anopheles mosquito bites a person, it injects sporozoites into the blood. Sporozoites infective stage for humans. Step 2. Liver stage. The sporozoites first enter the liver cells and multiply. Step 3. RBC stage. The parasites then enter the red blood cells, RBCs, and multiply. Step 4. Rupture of RBCs. The infected RBCs rupture and release new parasites, a toxic substance called hemazoin. Step 5. Fever and chills. The release of hemazoin causes chills, high fever, repeated fever attacks. The fever may recur at intervals of approximately 3 to 4 days, depending on the Plasmodium species. Step 6. Transmission back to mosquito. When a female Anopheles mosquito bites an infected person, it takes up the parasite. The parasite undergoes further development inside the mosquito and forms sporozoites in its salivary glands. The mosquito then transmits the parasite to another person. Neat important point. Parasite Plasmodium. Vector, female Anopheles mosquito, infective stage to humans, sporozoites, toxic substance hemozoin, hemozoin released during RBC rupture, most dangerous species, P. falciparum, hosts required, human and mosquito. Number 5, amoebiasis, amoebic dysentery, causative organism Embla histolytica, site of infection. The parasite lives in the large intestine. Transmission, contaminated food, contaminated water, houseflies. Role of housefly. The housefly acts as a mechanical carrier. It carries the parasite from the feces of an infected person to food and food products. Symptoms: constipation, abdominal pain, abdominal cramps, stool containing excess mucus, blood clots in stool. Neat point amoebiasis, Entamoeba histolytica, large intestine, housefly as mechanical carrier. Number 6, ascariosis, causative organism, Ascaris lumbricoides. It is a roundworm and an intestinal parasite. Transmission. The eggs of the parasite are released in the feces of infected people and contaminate soil, water, vegetables, fruits. A healthy person becomes infected by consuming contaminated food or water. Symptoms: internal bleeding, muscular pain, fever, anemia, blockage of intestinal passage. Neat point ascariosis, Ascaris lumbricoides, contaminated food and water. Number 7, filariasis elephantiasis. Causative organisms: Wuchereria bancrofti, Wuchereria malayi. These are filarial worms. Transmission: transmitted by the bite of infected female mosquito vectors. What happens? The worms cause chronic inflammation of the lymphatic vessels. Commonly affected areas: lower limbs, genital organs. Major symptom: severe swelling and enlargement of body parts, especially the lower limbs, resulting in elephantiasis. Neat point filariasis, Wuchereria, female mosquito, lymphatic vessels, elephantiasis. Number 8, ringworm. Ringworm is a fungal infection despite its name. It is not caused by a worm. Causative fungi: Microsporum, Trichophyton, Epidermophyton. Affected areas: skin, nails, scalp. Symptoms: dry lesions, scaly patches, itching. Favorable conditions: fungi grow well in warm conditions, moist conditions, poor personal hygiene. Neat point ringworm, fungi, dry, scaly lesions, skin, nails, and scalp. Quick neat revision table: disease, causative organism, type, transmission, important point. Typhoid. Salmonella typhi, bacterium, contaminated food and water, pneumonia, Streptococcus pneumoniae, H. influenzae, bacteria, respiratory droplets, common cold, rhinovirus, virus, droplets and contaminated objects, malaria, Plasmodium protozoan, female Anopheles mosquito, amoebiasis, Entamoeba histolytica, protozoan, contaminated food water, housefly, ascariosis, Ascaris lumbricoides, helminth, contaminated food water, filariasis, Wuchereria, helminth, female mosquito, ringworm, Microsporum, Trichophyton, Epidermophyton, fungi, direct contact and contaminated articles. Most important neat one-liners: 1. Typhoid is caused by Salmonella typhi. 2. Weedle test is used for typhoid diagnosis. 3. Pneumonia affects the alveoli. 4. Rhinovirus causes common cold. 5. Rhinovirus generally does not infect the lungs. 6. Female Anopheles mosquito transmits malaria. 7. Sporozoites are the infective stage of Plasmodium for humans. 8. Hemozoin causes recurring fever and chills in malaria. 9. Plasmodium falciparum causes malignant malaria. 10. Entamoeba histolytica causes amoebiasis. 11. Housefly acts as a mechanical carrier of amoebiasis. 12. Ascaris lumbricoides causes ascariasis. 13. Wuchereria causes filariasis or elephantiasis. 14. Ringworm is caused by fungi, not worms. 15. Microsporum, Trichophyton, and Epidermophyton cause ringworm. These organisms, symptoms, modes of transmission, vectors, and diagnostic tests are especially important for neat questions based directly on NCERT. Immunity. Deep explanation for neat immunity is the ability of the body to recognize foreign substances, antigens, and protect itself against disease-causing organisms and other harmful substances. The human body has a highly developed immune system consisting of cells, tissues, organs, and molecules that protect us from pathogens such as bacteria, viruses, fungi, protozoans, and helminths. Number 1. Types of immunity. Immunity is broadly divided into A. Innate immunity, present from birth, non-specific, provides immediate protection. B. Acquired adaptive immunity, develops during life, pathogen-specific, has immunological memory. A. Innate immunity. Innate immunity is the first line of defense against pathogens. It is present from birth and does not depend on previous exposure to a particular pathogen. 4 types of barriers in innate immunity. 1. Physical barriers. These prevent the entry of pathogens. Skin. The skin forms a protective physical barrier against microorganisms. Mucus coating. The mucus lining of the respiratory, gastrointestinal, and urogenital tracts traps microbes and prevents their entry into deeper tissues. 2. Physiological barriers. These are chemical conditions that destroy or inhibit pathogens. Examples: acid in the stomach kills many microorganisms. Lysozyme in tears and saliva destroys bacterial cell walls. Body temperature helps inhibit the growth of certain pathogens. Neat point lysozyme, tears, saliva, and nasal secretions, antibacterial enzyme. 3. Cellular barriers. Certain cells destroy pathogens by engulfing them through phagocytosis. Important cells: polymorphonuclear leukocytes, PMNLs, monocytes, macrophages, natural killer NK cells, natural killer cells. NK cells destroy virus-infected cells and tumor cells. Cytokine barriers. Virus-infected cells produce interferons. Interferons protect nearby healthy cells from viral infection. Neat point: virus-infected cells, interferons, protection of nearby healthy cells. B. Acquired adaptive immunity. Acquired immunity is developed during an individual's lifetime. It has 2 important properties. 1. Specificity. The immune system can distinguish between different pathogens. For example, the immune response against the measles virus is different from the response against the typhoid bacterium. 2. Memory. After the first exposure to a pathogen, the immune system remembers it. If the same pathogen enters the body again, the immune system produces a faster and stronger response. Primary and secondary immune response. Primary response. When a pathogen enters the body for the first time, the immune response is slow. The immune response is weak. Antibodies are produced gradually. This is called the primary immune response. Secondary response. When the same pathogen enters again, memory cells recognize it immediately. The response is faster. The response is stronger. A much larger amount of antibodies is produced. This is called the secondary immune response. Neat important: primary response equals slow and weak. Secondary response rapid and strong due to memory cells. This principle is the basis of vaccination and immunization. Cells involved in acquired immunity. The major cells involved are: 1. B lymphocytes. B cells produce antibodies. 2. T lymphocytes. T cells are mainly involved in cell-mediated immunity. They help destroy infected cells and regulate immune responses. Humoral immunity. Humoral immunity is mediated by antibodies present in body fluids, such as blood, lymph. It is mainly associated with B lymphocytes. When B cells encounter a specific antigen: 1. B cells become activated. 2. They form plasma cells. 3. Plasma cells produce antibodies. 4. Some B cells become memory cells. Antibodies. Antibodies are specialized proteins called immunoglobulins. They are produced by plasma cells. Basic structure. An antibody is made up of 2 heavy chains, 2 light chains. The antibody has antigen binding sites, a specific shape that binds to a specific antigen. This is often compared to a lock and key mechanism. Major types of immunoglobulins. Immunoglobulin, important role, IgG, most abundant antibody in blood. IgA, present in secretions such as saliva, tears, and breast milk. IgM, first antibody produced during primary immune response. IgE, associated with allergies. IgD, present on the surface of B cells. Neat focus, IgA, colostrum and secretions. IgE, allergy. IgM, first antibody produced. IgG, most abundant antibody in blood. Cell-mediated immunity. Cell-mediated immunity is mainly carried out by T lymphocytes. It is particularly important in destruction of virus-infected cells, rejection of transplanted organs, defense against intracellular pathogens, tumor immunity. Important T cell types: helper T cells, help activate other immune cells. Cytotoxic T cells, destroy infected or abnormal cells. Memory T cells, remember the pathogen and provide a faster response during future exposure. Active and passive immunity. Acquired immunity can also be classified as: 1. Active immunity. The person's own immune system produces antibodies. Natural active immunity develops after natural infection. Example: a person gets an infection, recovers, and develops immunity. Artificial active immunity develops after vaccination. Example, a vaccine introduces a harmless form or component of a pathogen, stimulating the body to produce antibodies and memory cells. Important features: develops slowly, long-lasting, produces memory cells. 2. Passive immunity. Ready-made antibodies are transferred to a person. Natural passive immunity antibodies are transferred from mother to child. Examples, IgG antibodies cross the placenta. IgA antibodies are present in colostrum. Artificial passive immunity. Ready-made antibodies are injected into the body. Example, antibodies given after exposure to certain toxins or infections. Important features: provides immediate protection, temporary. Memory cells are generally not formed. Active versus passive immunity. Feature: active immunity, passive immunity. Source of antibodies: body produces them. Ready-made antibodies received: speed, slow, immediate, duration, long-lasting, short-lived. Memory cells formed, generally not formed. Example, vaccination. Antibodies from colostrum. Vaccination and immunization. A vaccine contains killed microorganisms, weakened microorganisms, antigens, toxoids, components of pathogens. When a vaccine is administered, 1. Antigens stimulate the immune system. 2. Specific antibodies are produced. 3. Memory cells are formed. 4. On future exposure to the actual pathogen, a rapid secondary response occurs. Important concept: vaccination does not usually provide immediate immunity. It prepares the immune system for future infection. Colostrum and immunity. Colostrum is the first milk produced by the mother after childbirth. It contains a high concentration of IgA antibodies. These antibodies provide passive immunity to the newborn. Neat point colostrum, IgA, passive immunity in newborn. Maternal antibodies. Through placenta, IgG antibodies can cross the placenta from the mother to the fetus. This provides passive immunity to the developing fetus. Neat point placenta IgG transfer, colostrum IgA transfer. Important concept: antigen and antibody. Antigen. An antigen is a foreign substance that stimulates an immune response. Examples: proteins on the surface of bacteria, viral particles, toxins. Antibody. An antibody is a specific protein produced by the immune system in response to an antigen. Simple sequence: antigen enters, B cells activated, plasma cells formed, antibodies produced. Innate versus acquired immunity. Feature: innate immunity, acquired immunity. Present from birth, develops during life. Specificity: nonspecific, highly specific. Memory: absent. Present response: immediate, slow during first exposure. Main components: skin, mucus, phagocytes, interferons, B cells, T cells, antibodies. Neat most important points. 1. Innate immunity is present from birth. 2. Acquired immunity is specific and has memory. 3. B lymphocytes produce antibodies. 4. T lymphocytes are involved in cell-mediated immunity. 5. NK cells destroy virus-infected and tumor cells. 6. Interferons are produced by virus-infected cells. 7. Lysozyme is present in tears, saliva, and nasal secretions. 8. IgM is the first antibody produced during the primary immune response. 9. IgG is the most abundant antibody in blood. 10. IgA is present in colostrum and secretions. 11. IgE is associated with allergic reactions. 12. IgG crosses the placenta. 13. GA in colostrum provides passive immunity to newborns. Vaccination produces active immunity. 15. Secondary immune response is faster and stronger than primary response. 16. Memory cells are responsible for long-term immunological memory. 17. Passive immunity gives immediate but temporary protection. Neat memory trick game for antibody classes. GIGG goes through placenta. A. IgA available in secretions and colostrum. M. IgM made first. E. IgE eosinophils and allergy. D. IgD development surface of B cells. Allergies. Neat second PUC biology. Allergy is an exaggerated or inappropriate immune response of the body to certain normally harmless substances called allergens. These substances are usually harmless to most people, but the immune system of an allergic person recognizes them as foreign and produces an excessive immune response. 1. What are allergens? Allergens are substances that trigger an allergic reaction. Common allergens include dust, pollen grains, house dust mites, animal dander, certain foods, insect bites, drugs and medicines, cosmetics, certain chemicals. Neat point allergen, unwanted, exaggerated immune response, allergy. Number 2, mechanism of allergy. The important antibody involved in allergy is IgE. When an allergen enters the body, step 1, first exposure. The allergen stimulates the immune system. Step 2, IgE production. The body produces IgE antibodies against that allergen. Step 3, IgE attaches to cells. IgE antibodies bind to the surface of mast cells, basophils. Step 4, repeated exposure. When the same allergen enters the body again, it interacts with IgE antibodies on mast cells. Step 5, chemical release. Mast cells release chemicals such as histamine, serotonin, other inflammatory mediators. Step 6, symptoms appear. These chemicals produce the symptoms of allergy. Simple flowchart: allergen, IgE production, IgE binds mast cells, re-exposure, histamine release, allergy symptoms. Number 3, symptoms of allergy. Common symptoms include sneezing, running nose, nasal congestion, coughing, watery eyes, redness of eyes, itching, skin rashes, swelling, difficulty in breathing. In severe cases, a serious allergic reaction called anaphylaxis can occur. Number 4, examples of allergic conditions. 1, allergic rhinitis, caused by allergens such as pollen, dust mites. Symptoms: sneezing, running nose, itchy nose, watery eyes. 2, asthma. Some forms of asthma may be triggered by allergens. Symptoms include difficulty in breathing, wheezing, chest tightness. 3, skin allergy. May cause itching, redness, rashes, swelling. 5, diagnosis of allergy. Allergy can be diagnosed using skin tests, blood tests, allergy testing. In a skin test, a small amount of a suspected allergen is introduced into the skin. If the person is allergic to that substance, an immune reaction occurs at the site. Number 6, treatment of allergy. According to NCERT, allergy is treated using antihistamines. They block the effects of histamine. Adrenaline, used in severe allergic reactions. Steroids, used to reduce inflammation. Important concept. Treatment aims to 1, reduce the allergic response. 2, control symptoms. 3, prevent exposure to the allergen whenever possible. Number 7, allergy and IgE. Important neat connection. Component, role, allergen. Triggers allergic reaction, IgE, main antibody involved. Mast cells release inflammatory chemicals. Histamine produces allergy symptoms. Antihistamines reduce histamine effects. Number 8, allergy versus normal immune response. Normal immune response, allergic response, protects the body from harmful pathogens, occurs against harmless substances. Controlled response, exaggerated response, helps eliminate infection, causes symptoms and inflammation. Usually beneficial, may be harmful. Neat most important points. 1, allergy is an exaggerated immune response. 2, substances causing allergy are called allergens. 3, IgE antibodies are mainly involved in allergic reactions. 4, mast cells release histamine during allergy. 5, histamine and serotonin are important chemicals involved in allergic reactions. 6, common allergens include dust, pollen, mites, and animal dander. 7, antihistamines, adrenaline, and steroids are used to treat allergy. 8, anaphylaxis is a severe and potentially life-threatening allergic reaction. 9, the first exposure to an allergen leads to sensitization and IgE production. 10, repeated exposure to the same allergen causes mast cell degranulation and release of histamine. Neat memory trick. AIMH. A, allergen enters. I, IgE is produced. M, mast cells are activated. H, histamine is released. Allergen, IgE. HIV infection. Complete neat explanation. AIDS is an important topic from human health and diseases. For NEAT, you should understand the virus, target cells, replication mechanism, transmission, symptoms, diagnosis, treatment, and prevention. The NCERT mechanism of HIV infection is especially important. 1. Full forms. HIV, human immunodeficiency virus. AIDS, acquired immunodeficiency syndrome. ART, antiretroviral therapy. CD4, cluster of differentiation 4. ELISA, enzyme-linked immunosorbent assay. NACO, National AIDS Control Organization. Meaning of AIDS: acquired, not present from birth, acquired during life. Immuno, related to the immune system. Deficiency, reduced ability of the immune system. Syndrome, a collection of signs and symptoms. Therefore, AIDS is a condition in which the immune system becomes severely weakened, making the person vulnerable to many infections. Number 2, causative agent. HIV/AIDS is caused by HIV, which is a retrovirus. Why is HIV called a retrovirus? HIV contains RNA as genetic material, normally DNA, RNA, protein, but in HIV, RNA, DNA. This reverse process is carried out by the enzyme reverse transcriptase. Therefore, HIV is called a retrovirus. Number 3, important structure of HIV. HIV contains 1 RNA genome, 2 reverse transcriptase enzyme, 3 protein capsid, 4 lipid envelope, 5 surface glycoproteins that help the virus attach to host cells. Neat point open quote, HIV is an enveloped RNA virus and contains reverse transcriptase. Close quote. Number 4, main target cells of HIV. HIV mainly attacks helper T lymphocytes, CD4 to my T cells. These cells are extremely important because they coordinate the immune response. They help activate B lymphocytes, antibody production, cytotoxic T cells, destruction of infected cells, other immune cells. Therefore, HIV infection decrease in CD4 mice helper T cells, weakening of immunity. This is the central concept of AIDS. Number 5, mechanism of HIV infection. Very important for NEAT. Let us understand the process step by step. Step 1, entry into the body. HIV enters the human body through infected body fluids. The virus initially infects macrophages. Step 2, viral RNA enters the macrophage. HIV releases its RNA and enzymes into the host cell. Step 3, reverse transcription. The enzyme reverse transcriptase converts viral RNA, viral DNA. This is a very important NEAT point. Step 4, integration into host DNA. The viral DNA becomes integrated with the DNA of the host cell. This allows the virus to use the host cell machinery to produce new viral components. Step 5, formation of new viruses. The infected cell produces viral RNA, viral proteins, new viral particles. These combine to form new HIV particles. Step 6, infection of helper T cells. HIV also enters helper T lymphocytes. Inside these cells, the virus replicates. The newly formed viruses are released and infect other healthy helper T cells. Step 7, progressive decrease in T cells. As the number of helper T cells gradually decreases, helper T cells, immune response, ability to fight infections, opportunistic infections, AIDS. This mechanism is directly based on the NCERT explanation and is one of the most important areas for NEAT questions. Number 6, what are opportunistic infections? When immunity becomes severely weakened, microorganisms that normally may not cause serious disease can cause severe infections. These are called opportunistic infections. Examples may include serious bacterial infections, viral infections, fungal infections, protozoan infections. Why do they occur? Because HIV destroys the immune system's ability to properly control infections. Number 7, modes of transmission of HIV. HIV is transmitted through certain infected body fluids. 1, sexual contact. Unprotected sexual contact with an infected person can transmit HIV. 2, infected blood transfusion. Transfusion of infected blood can transmit HIV. Therefore, donated blood must be properly screened. 3, sharing contaminated needles and syringes. Sharing infected needles can transmit HIV. This is particularly important in people who share injection equipment. 4, mother-to-child transmission. An infected mother can transmit HIV to her child during pregnancy, during childbirth, through breastfeeding. This is called vertical transmission or mother-to-child transmission. NEAT trap. HIV is not transmitted by handshaking, hugging, casual touching, sharing food, sharing a classroom, sitting beside an infected person, mosquito bites. The virus requires exposure to infected body fluids, not ordinary social contact. Number 8, symptoms of AIDS. The symptoms develop because the immune system becomes progressively weaker. Common manifestations may include persistent fever, weight loss, chronic diarrhea, recurrent infections, severe weakness, enlarged lymph nodes, opportunistic infections. The important idea is, open quote, the person becomes vulnerable to infections that a healthy immune system would normally control, close quote. Number 9, diagnosis of HIV infection. ELISA. The commonly taught NCR test for HIV infection is ELISA, enzyme-linked immunosorbent assay. It is used to detect evidence of HIV infection. NEAT memory point, HIV ELISA. Marten, treatment of AIDS. At present, there is no simple complete cure that eliminates HIV from the body. However, HIV can be controlled with antiretroviral therapy, ART. ART reduces viral replication, lowers the amount of virus in the blood, helps preserve immune function, can greatly improve lifespan and quality of life. Modern treatment can suppress HIV very effectively, but treatment does not mean the virus has been completely eliminated. Number 11, prevention of AIDS. Prevention is extremely important. Important measures. 1, practicing safe sex. 2, avoiding sharing of needles and syringes. 3. Screening blood before transfusion. 4. Using sterile medical equipment. 5. Testing and treatment during pregnancy when appropriate. 6. Creating awareness. 7. Avoiding stigma and discrimination. NCERT concept, open quote, prevention of AIDS is the most important strategy, close quote. 12. AIDS versus HIV. HIV/AIDS, human immunodeficiency virus, acquired immunodeficiency syndrome. It is a virus. It is a disease condition syndrome. Causes HIV infection, advanced stage of HIV infection, attacks immune cells, results in severe immune deficiency. Chash easy memory trick. HIV, cause AIDS, result. 13. Complete flowchart for NEAT. You can see the code in our conversation history. Most important NEAT facts. 1. AIDS is caused by HIV. 2. HIV is a retrovirus. 3. HIV contains RNA. 4. Reverse transcriptase converts RNA into DNA. 5. Main target cells are helper T lymphocytes. 6. Macrophages are also infected by HIV. 7. Decrease in T helper cells causes immune deficiency. 8. ELISA is associated with HIV diagnosis. 9. Infected blood can transmit HIV. 10. Sharing contaminated needles can transmit HIV. 11. Mother-to-child transmission is possible. 12. Casual touch does not transmit HIV. 13. AIDS is a syndrome, not the name of the virus. 14. AART helps control AV infection. 15. Opportunistic infections occur due to weakened immunity. NEAT style questions. Q1. AIDS is caused by A. Bacteria, B. Retrovirus, C. Protozoan, D. Fungus. Answer B. Retrovirus. HIV is an RNA-containing retrovirus. Q2. The enzyme responsible for the formation of DNA from RNA in HIV is DNA polymerase, B. RNA polymerase, C. Reverse transcriptase, D. Ligase. Answer C. Reverse transcriptase. Q3. HIV primarily attacks ARBCs, B. Helper T lymphocytes, C. Platelets, D. Neutrophils only. Answer B. Helper T lymphocytes. Q4. The correct sequence in HIV replication is a DNA, RNA, dRNA, DNA, viral proteins. C. Protein, RNA, DNA. D. DNA, protein, RNA. Answer B. RNA, DNA, viral proteins. Which of the following is not a mode of HIV transmission? A. Infected blood transfusion. B. Sharing infected needles. C. Unprotected sexual contact. D. Handshaking. Answer D. Handshaking. Q6. A person infected with HIV becomes susceptible to many infections because A. RBC count increases. B. Helper T cell number decreases. C. Platelets are destroyed. D. Antibodies become permanently absent immediately. Answer B. Helper T cell number decreases. Q7. ELISA is commonly associated with the diagnosis of A. Malaria, B. AIDS HIV infection, C. Typhoid, D. Ringworm. Answer B. AIDS HIV infection. Q8. HIV is called a retrovirus because A. It contains only DNA. B. It produces RNA from protein C. It forms DNA from RNA using reverse transcriptase D. It lacks genetic material. Answer C. It forms DNA from RNA using reverse transcriptase. Q9. Which of the following can transmit HIV? A. Sharing food. B. Mosquito bite. C. Transfusion of infected blood. D. Handshake. Answer C. Transfusion of infected blood. Q10. The major cause of immune deficiency in AIDS is A. Increase in RBCs. B. Destruction, reduction of helper T cells. C. Increase in platelets. D. Increase in body temperature only. Answer B. Destruction, reduction of helper T cells. Assertion reason question. Assertion A. HIV-infected individuals become susceptible to opportunistic infections. Reason R. HIV progressively decreases the number of helper T lymphocytes. Answer. Both A and R are true, and R is the correct explanation of A. NEAT match the following. Column 1, Column 2, HIV, retrovirus. Reverse transcriptase, RNA, DNA, helper T cell, immune coordination, HIV diagnosis, ART control of HIV infection. Correct matching. HIV retrovirus, reverse transcriptase RNA to DNA, helper T cell, immune coordination. ELISA HIV diagnosis. ART control of HIV infection. 1 minute revision. HIV, RNA virus, reverse transcriptase, DNA, host DNA, new viruses, T helper cell destruction, immunity decreases, opportunistic infections, AIDS. This single chain is the most important concept to remember for NEAT. NEAT strategy. Read the AIDS portion directly from NCERT, especially the paragraphs on HIV transmission and then the mechanism by which HIV causes immune deficiency. The official NCERT text includes these as direct exercise targets. Cancer. Complete NEAT explanation. Cancer is a very important topic from Class 12 Biology, Human Health and Diseases. For NEAT, focus especially on uncontrolled cell division, benign versus malignant tumors, metastasis, carcinogens, oncogenes, diagnosis, treatment, and prevention. 1. What is cancer? Cancer is a disease caused by uncontrolled and abnormal division of cells. Normally, cells divide when required, stop dividing when enough cells are present, undergo programmed cell death when damaged or unnecessary. In cancer, this control is lost. Normal condition: cell growth, cell division, cell death when necessary. Cancerous condition: uncontrolled division, abnormal cell accumulation, tumor formation, possible spread. Cancer is fundamentally a disease involving genetic changes that disrupt normal control of cell growth, division, and death. Number 2. What is a tumor? A tumor is an abnormal mass of cells produced due to uncontrolled cell division. Tumors are mainly of two types. Benign tumor. A benign tumor usually remains localized, does not invade nearby tissues, does not spread to distant organs, is generally less dangerous. Example: a localized tumor that grows slowly and remains at its original site. B. Malignant tumor. A malignant tumor grows rapidly, invades surrounding tissues, can enter blood or lymphatic vessels, can spread to distant organs, is cancerous. Important neat point, open quote. Malignant tumor equals cancerous tumor, close quote. The ability of malignant cancer cells to invade other tissues and form new tumors at distant locations is called metastasis. Number 3. Benign versus malignant tumor. Benign tumor, malignant tumor. Growth: usually slow, often rapid. Local invasion, absent or limited. Present. Spread: does not metastasize, can metastasize. Nature: usually non-cancerous. Cancerous: danger: usually less dangerous, potentially life-threatening. Hash, easy memory trick. Benign equal bounded malignant. Move 4. What is metastasis? Very important for NEAT. Metastasis is the spread of cancer cells from the original site to distant parts of the body. Example: cancer begins in the breast. Cancer cells travel to the lung. The tumor in the lung is still metastatic breast cancer, not lung cancer, because the cancer cells originated from the breast. Cancer cells may spread through blood vessels, lymphatic vessels. Basic sequence: you can see the code in our conversation history. The new tumor is called a secondary tumor or metastatic tumor. Number 5. How does cancer develop? Cancer develops due to changes in genes controlling cell division, cell growth, DNA repair, programmed cell death. Normally, damaged cells may be repaired or destroyed, but if mutations accumulate in important genes, the cell may become cancerous. Flowchart: you can see the code in our conversation history. Number 6. Cell cycle in cancer. The cell cycle is normally controlled by growth signals, cell cycle checkpoints, tumor suppressor genes, DNA repair mechanisms, apoptosis. Cancer occurs when these control mechanisms fail. Normal cell: you can see the code in our conversation history. Cancer cell: you can see the code in our conversation history. Number 7. Oncogenes, very important. Proto-oncogenes. Proto-oncogenes are normal genes that help regulate cell growth, cell division, cell survival. If a proto-oncogene undergoes certain mutations or becomes excessively active, it can become an oncogene. An oncogene promotes uncontrolled cell growth. Remember, you can see the code in our conversation history. Neat question: a normal gene that becomes cancer-causing after mutation is called answer, proto-oncogene, oncogene. Cancer-related genetic changes also involve tumor suppressor genes and DNA repair genes. Number 8. Tumor suppressor genes. Tumor suppressor genes act like brakes on cell division. They can stop uncontrolled cell division, promote DNA repair, trigger apoptosis in severely damaged cells. If tumor suppressor genes become inactive, you can see the code in our conversation history. Easy analogy: proto-oncogene equals accelerator. Tumor suppressor gene equal brake. Cancer can occur when the accelerator becomes permanently active or the brake stops working. Number 9. DNA repair genes. DNA repair genes correct damage in DNA. If these genes are defective, you can see the code in our conversation history. Thus, 3 important gene groups are: 1. Proto-oncogenes. Promote normal growth. Abnormal activation can cause cancer. 2. Tumor suppressor genes restrict abnormal growth. Loss of function can cause cancer. 3. DNA repair genes repair DNA damage. Defects allow mutations to accumulate. 10. Causes of cancer. Cancer may result from a combination of genetic mutations, environmental factors, lifestyle factors, certain infections, radiation, chemical exposure. Number 11. Carcinogens. A carcinogen is an agent that can cause cancer. Carcinogens may be A. Physical, X-rays, gamma rays, ultraviolet radiation. B. Chemical. Tobacco smoke, certain industrial chemicals, some chemical pollutants. C. Biological. Certain cancer-causing viruses are called oncogenic viruses. Number 12. Tobacco and cancer. Tobacco smoking is strongly associated with several cancers, especially lung cancer, oral cancer, throat cancer. Tobacco smoke contains cancer-causing substances, NEAT concept smoking, carcinogens, DNA damage, mutations, cancer risk. Number 13. Radiation and cancer. Excessive exposure to radiation can damage DNA. Examples: X-rays, gamma rays, ultraviolet radiation. UV radiation is particularly associated with DNA damage in skin cells, increased risk of skin cancer. Number 14. Oncogenic viruses. Some viruses can contribute to cancer development. These viruses may introduce genetic changes or interfere with normal cell cycle control. Important examples: human papillomavirus, HPV, associated with cervical and other cancers. Hepatitis B virus, HBV, associated with liver cancer. Hepatitis C virus, HCV, associated with liver cancer. Epstein-Barr virus, EBV, associated with certain lymphomas and other cancers. HTLV-1, associated with adult T-cell leukemia lymphoma. For NEAT, remember the general concept. Open quote. Some viruses can act as oncogenic viruses and contribute to cancer development. Close quote. Number 15. Characteristics of cancer cells. Cancer cells differ from normal cells. They may, 1. Divide uncontrollably. They continue dividing even when normal cells would stop. 2. Ignore growth-inhibiting signals. Normal cells respond to signals that stop division. Cancer cells may ignore these signals. 3. Avoid apoptosis. Normally, severely damaged cells undergo programmed cell death. Cancer cells may avoid this process. 4. Invade surrounding tissues. Malignant cells can penetrate nearby tissues. 5. Metastasized cancer cells can spread to distant organs. 6. Stimulate new blood vessel formation. A growing tumor requires oxygen, nutrients. Therefore, tumor cells can stimulate the formation of new blood vessels. This process is called angiogenesis. 7. Escape immune destruction. Some cancer cells can avoid recognition or destruction by the immune system. These features explain why cancer cells can continue growing and spreading. Number 16. Primary tumor and secondary tumor. Primary tumor, the tumor at the original site of cancer development. Secondary tumor, a tumor formed at another site after metastasis. Example: you can see the code in our conversation history. The brain tumor is a secondary tumor from lung cancer. Number 17. Symptoms of cancer. Symptoms depend on type of cancer, organ affected, size of tumor, stage of disease, whether metastasis has occurred. Possible symptoms include unexplained weight loss, persistent fatigue, unusual lump or swelling, abnormal bleeding, persistent cough, change in bowel or bladder habits, non-healing wound, unexplained pain. Important: these symptoms do not automatically mean cancer. Proper medical evaluation is required. Number 18. Diagnosis of cancer. Cancer diagnosis may involve several methods. A. Biopsy. A small sample of tissue is removed and examined under a microscope. Neat point biopsy equals removal and microscopic examination of tissue. A biopsy is often the definitive way to determine whether a suspicious lesion is cancerous. B. Imaging techniques. Examples: X-ray, CT scan, MRI, ultrasound, PET scan. These can help identify tumors, their size, their location, possible spread. C. Blood and laboratory tests. Blood and other body fluid tests can help detect abnormalities and tumor markers. However, abnormal laboratory results alone do not necessarily prove cancer. D. Molecular and genetic testing. Modern cancer diagnosis may examine genetic mutations, biomarkers, molecular features of tumor cells. Diagnosis usually involves combining physical examination, laboratory tests, imaging, and often biopsy. Number 19. Cancer staging. Cancer staging describes size of the primary tumor, involvement of nearby tissues, lymph node involvement, distant metastasis. General concept: you can see the code in our conversation history. Important: metastasis generally indicates advanced disease. Number 20. Treatment of cancer. Treatment depends on type of cancer, stage, location, genetic features, overall health of the patient. Common treatments include A. Surgery. The tumor is physically removed, most useful when the tumor is localized and can be safely removed. B. Radiotherapy. High-energy radiation is used to damage and kill cancer cells. Important concept. Cancer cells are often more sensitive to radiation than many normal cells, but surrounding normal tissues can also be affected. C. Chemotherapy. Anti-cancer drugs are used to kill or inhibit rapidly dividing cells. Possible side effects. Because some normal cells also divide rapidly, chemotherapy may affect hair follicles, bone marrow, digestive tract cells. This can result in hair loss, reduced blood cell production, nausea. D. Immunotherapy. The immune system is helped or activated to attack cancer cells. Example concept. Immune cells may be encouraged to better recognize and destroy tumor cells. E. Targeted therapy. These treatments target specific molecular changes that help cancer cells grow or survive. F. Hormone therapy. Some cancers depend on hormones for growth. Hormone therapy can reduce or block these hormonal signals. Modern cancer treatment may use one treatment alone or combinations such as surgery, chemotherapy, and radiation therapy depending on the cancer and its stage. Number 21. Cancer immunotherapy and biological response modifiers. A very important NSAID-related concept is the use of substances that help the immune system fight cancer. Interferons. Interferons are biological response modifiers that can help enhance immune responses against cancer cells. Monoclonal antibodies. They may be designed to recognize specific molecules on cancer cells. Number 22. Prevention of cancer. Important preventive measures include 1. Avoid tobacco. Do not smoke or use tobacco products. 2. Avoid unnecessary radiation exposure. Radiation should be used appropriately and safely. 3. Protect skin from excessive UV radiation. Avoid excessive exposure to strong sunlight. 4. Maintain a healthy lifestyle. Important factors include balanced diet, regular physical activity, healthy body weight, avoiding tobacco, limiting harmful exposures. 5. Screening. Screening can help detect some cancers at an earlier stage. Early detection can improve treatment options. Number 23. Cancer versus tumor. This is a common confusion. Tumor, an abnormal mass of cells. Cancer, a malignant disease involving uncontrolled growth and potentially invasion and metastasis. Therefore, open quote, every cancerous solid tumor is a tumor, but not every tumor is cancerous. Close quote. Number 24. Cancer and apoptosis. Normal situation. You can see the code in our conversation history. Cancer situation. You can see the code in our conversation history. Neat point failure of programmed cell death can contribute to cancer. Number 25. Cancer and contact inhibition. Normal cells generally stop dividing when they come into contact with neighboring cells. This is called contact inhibition. Cancer cells may lose contact inhibition. Therefore, you can see the code in our conversation history. This contributes to tumor formation. 26. Angiogenesis. A tumor cannot grow indefinitely without a blood supply. Cancer cells can stimulate nearby blood vessels to grow toward the tumor. This process is angiogenesis. Why is it important? New blood vessels provide oxygen, nutrients, waste removal. Thus, angiogenesis helps tumor growth. Number 27. Complete cancer mechanism. You can see the code in our conversation history. Most important neat facts. 1. Cancer is uncontrolled cell division. 2. Malignant tumors are cancerous. 3. Benign tumors usually remain localized. 4. Metastasis is the spread of cancer cells to distant organs. 5. Proto-oncogene can become an oncogene. 6. Tumor suppressor genes act as brakes on cell division. 7. Carcinogens can cause cancer. 8. Tobacco smoke contains carcinogenic substances. 9. Radiation can damage DNA and cause cancer. 10. Biopsy is an important diagnostic method. 11. Radiotherapy uses radiation. 12. Chemotherapy uses anti-cancer drugs. 13. Immunotherapy helps the immune system fight cancer. 14. Angiogenesis supplies blood to a growing tumor. 15. Cancer cells may avoid apoptosis. 16. Cancer cells may lose contact inhibition. 17. Oncogenic viruses can contribute to cancer. 18. Metastatic cancer retains the type of the primary cancer. 19. Cancer is fundamentally associated with genetic changes. Early detection and prevention are extremely important. Hash neat practice questions. Q1. A tumor that can invade surrounding tissues and spread to distant organs is A. Benign tumor, B. Malignant tumor, C. Cyst, D. Hyperplastic tissue. Answer B. Malignant tumor. Q2. The spread of cancer cells from the original site to distant organs is called A. Apoptosis, B. Metastasis, C. Angiogenesis, D. Necrosis. Answer B. Metastasis. Q3. A normal gene that may become cancer-causing after mutation is A. Anticodon, B. Proto-oncogene, C. Structural gene only, D. Ribosomal gene. Answer B. Proto-oncogene. Q4. Tumor suppressor genes normally promote uncontrolled cell division. B. Help control cell growth and division. C. Destroy all RBCs. D. Produce viral RNA. Answer B. Help control cell growth and division. Q5. Which of the following is a carcinogen? A. Tobacco smoke. B. Oxygen under normal conditions. C. Water. D. Glucose. Answer A. Tobacco smoke. Q6. The formation of new blood vessels to supply a tumor is called A. Metastasis. B. Angiogenesis. C. Apoptosis. D. Phagocytosis. Answer B. Angiogenesis. Q7. Which technique involves microscopic examination of a tissue sample? A. Biopsy. B. Vaccination. C. Dialysis. D. ELISA only. Answer A. Biopsy. Q8. Cancer cells commonly show A. Strict control of cell division. B. Increased apoptosis. C. Uncontrolled cell division. D. Complete absence of DNA. Answer C. Uncontrolled cell division. Q9. Which treatment uses high-energy radiation? A. Chemotherapy. B. Radiotherapy. C. Immunotherapy. D. Hormone therapy. Answer B. Radiotherapy. Q10. A cancer that spreads from the breast to the lung is called A. Lung cancer. B. Metastatic breast cancer. C. Primary lung cancer. D. Benign lung tumor. Answer B. Metastatic breast cancer. Assertion, reason, question. Assertion. Malignant tumors are more dangerous than benign tumors. Reason. Malignant tumors can invade surrounding tissues and undergo metastasis. Answer. Both assertion and reason are true, and reason is the correct explanation of assertion. Match the following column 1, column 2. Proto-oncogene may become oncogene. Tumor suppressor gene controls abnormal growth. Biopsy, tissue examination, radiotherapy, high-energy radiation, metastasis, spread of cancer. 1-minute revision, mutation, oncogene activation, tumor suppressor failure, loss of cell cycle control, uncontrolled cell division, tumor, malignancy, invasion, metastasis. The most important NEAT concepts are, open quote, oncogene plus tumor suppressor genes, carcinogens, biopsy plus radiotherapy plus chemotherapy plus metastasis plus angiogenesis, close quote. For NEAT preparation, revise this topic along with the exact NCRT Human Health and Diseases section, because direct conceptual questions are commonly built from these definitions and mechanisms. Cancer is explicitly included in the official biology syllabus under human diseases.